Good day, everyone. My name is Adrin, and today I'm going to report about the next generation sequencing, Illumina. What is next generation sequencing? Next generation sequencing, or NGS, is a DNA sequencing technology that enables the complete sequencing of a human genome in a short span of time, given a single day. This method is more powerful and faster than the previous generation of sequencing, which required several decades to complete. It is a revolutionary technology that enables researchers to perform wide-scale analysis of biological systems at a speed never before possible. Its ability to provide high-quality, fast, and cost-effective results has made it an integral part for modern life. Now, let us learn the advantages of using next-generation sequencing. It rapidly sequences whole genomes. It deeply sequences target regions. It utilizes RNA sequencing to discover novel RNA variants and splice sites or quantify mRNAs for gene expression analysis. It analyzes epigenetic factors such as genome-wide DNA methylation and DNA-protein interactions. Sequences cancer samples to study rare somatic variants, tumor, subclones, and more. It studies the human microbiome and it identifies novel pathogens. Now let us learn the limitations of NGS. NGS in a clinical setting is putting into place the required infrastructure such as computer capacity and storage, and also the personal expertise required to comprehensively analyze and interpret the subsequent data. The volume of data needs to be managed skillfully to extract the clinically important information in a clear and robust interface. The actual sequencing cost of NGS is negligible. For example, a state-of-the-art NGS platform can generate approximately 150 million reads for around €1,000, whereas a single Sanger read typically costs less than €1. Now, to make the NGS cost-effective, one would have to run large batches of samples, which may require super-regional centralization. Following the initial capital investment, the capacity of an NGS facility can provide a service on national scale, likely offering economic benefits in addition to improvements in patient care. What is Illumina sequencing? The Illumina sequencing process differs significantly from the traditional Sanger chain termination method. It uses massively parallel sequencing by synthesis, or SBS technology, to track the insertion of tagged nucleotides as the DNA chain is duplicated. It can deliver data output ranging from 300 kilobases up to multiple terabases in a single run, depending on instrument type and configuration. It is a widely used NGS technology that accounts for more than 90% of all sequencing data generated worldwide. One Illumina sequencing tools and reagents support massively parallel sequencing. Through the years, Illumina has continuously innovated. In this part, we will learn the recent Illumina NGS breakthroughs. Number 1. Semiconductor sequencing with CMOS technology. The ISEQ100 system combines a complementary metal oxide semiconductor, or CMOS chip, with one-channel SBS to deliver high-accuracy data in a compact system. Number 2 is a two-channel SBS. This technology enables faster sequencing than the original four-channel versions of the SBS technology, with the same high data accuracy. Number 3 is a pattern flow cell technology. This option offers an exceptional level of throughput for diverse sequencing applications. Number 4, its ability to tune up output for up to 6 terabytes in estimatedly 2 days. Lastly, the 75 breakthrough innovations. The next SEQ 1000 and 2000 systems offer flexibility for emerging applications. It allows data analysis in as little as 2 hours. The following is the next generation sequencing workflow steps. Number 1, DNA isolation. Estimated time is about 1 to 2 hours. Number 2, library preparation. Estimated time is about 2.5 hours. Number 3, sequencing. Estimated runtime is about 19.5 hours. And lastly is data analysis, which is estimated time is about 1 hour. The first step is DNA isolation. In order to prepare your samples, you need to isolate and purify it first. For best results, use an extraction protocol optimized for the sample type. Isolate the DNA from microbial colonies without introducing inhibitors. Using glass beads is recommended. A UV spectrophotometry is used to assess purity, and to quantitate DNA, we use fluorometric methods. Estimated time is about 1 to 2 hours. The next step is library preparation. This procedure prepares DNA or RNA samples to be sequencer compatible. The most common method for creating sequencing libraries is to break DNA and add specific adapters to both ends. These adapters contain complementary sequences that allow DNA fragments to bind to the flow cell in the Illumina sequencing method. After that, fragments can be amplified and purified. Multiple libraries can be pulled together and sequenced in the same run to conserve resources. This is known as multiplexing. Unique index sequences or barcodes are inserted to each library during adapter ligation. Estimated time is about 2.5 hours, and estimated DNA input is from 1 to 500 ng. Now let us observe the diagram. The first step is fragmentation. Short-read sequencing technologies like those from Illumina cannot readily analyze very long DNA strands, so samples are fragmented into uniform pieces to make them amenable to be sequencing. After fragmentation, the DNA fragments are end repaired or end polished. This part here, and this part here. Generally, a single adenine base is added to form an overhang via an A-tailing reaction. This A overhang allows adapters containing a single thymine overhanging base to base pair with the DNA fragment. So this is called adenylation, the addition of an adenine base. Next is the ligation of adapters. A ligase enzyme is covalently linked to the adapter and inserts DNA fragments, making a complete library molecule. This part here, and this part here. These adapters serve multiple functions. They attach the sequences to the flow cell to allow sequencing, and then they contain barcodes that also called indexes to identify samples and permit multiplexing. The last part is the PCR amplification. This is an optional step though. Whether or not you amplify your libraries depends on the adapter type and sample input used. After PCR amplification, remaining oligonucleotides and small fragments must be removed. PCR cleanup can be performed using magnetic beads or a spin column. The third part is sequencing. Libraries are put into a flow cell and placed on the sequencer during the sequencing steps of the NGS method. Cluster creation is a mechanism that amplifies DNA fragment clusters, resulting in millions of copies of a single-stranded DNA. Clustering is done automatically on most Illumina sequencing devices. The chemically modified nucleotides bind to the DNA template strand by naturally complementarity in a process known as sequencing by synthesis or SBS. Each nucleotide has a fluorescent tag as well as a reversible terminator that prevents the following base from being incorporated. The terminator is cleaved so that the next base may bind, and the fluorescent signal reveals which nucleotide has been inserted. After reading the forward DNA strand, the reads are washed away and the process repeats for the reverse strand. This method is called the paired end sequencing. This part is estimated to have a runtime of 19.5 hours and an estimated output of 1.2 gigabytes per 2 times 150 base pair run. Samples per run is about 4 to 5 samples, assuming that 5 megabytes has 50 times coverage. Now that we are done sequencing, let's go on to the last part, the data analysis. This entire process generates millions of reads of all fragments. Sequences from pooled sample libraries are separated based on their unique indices introduced during sample preparation. For each sample, reads with similar stretches or base code are locally clustered. So in this diagram we show here that they are clustered differently and they are aligned to the original genome. And these contigs or the overlapping regions are the ones that they will actually look at. Now forward and reverse reads are paired, creating a contiguous sequence. These contiguous sequences are aligned to the reference genome of the variant identification. The paired end information is used to resolve ambiguous alignments. Now data analysis would have an estimated runtime of about 1 hour. In summary, next-generation sequencing, or NGS, is a DNA sequencing technology that enables the complete sequencing of a human genome in a short span of time. Illumina is a widely used NGS technology that uses massively parallel sequencing by synthesis or SBS technology to track the insertion of tagged nucleotides as the DNA chain is duplicated. The next generation sequencing workflow follows these steps: DNA isolation, library preparation, sequencing, and data analysis. This entire process generates millions of reads of all fragments. The following are the references used for this presentation. Thank you for listening.